getting results from a blood test can take anywhere from 1 day to a week, depending on what a test is targeting. The same goes for tests of water pollution and food contamination. And in most cases, the wait time has to do with time-consuming steps in sample processing and analysis. Now, MIT engineers have identified a new optical signature in a widely used class of magnetic beads, which could be used to quickly detect contaminants in a variety of diagnostic tests. For example, the team showed the signature could be used to detect signs of the food contaminant Salmonella. The so-called Dynabits are microscopic magnetic beads that can be coated with antibodies that bind to target molecules such as a specific pathogen. Dynabits are typically used in experiments in which they are mixed into solutions to capture molecules of interest. But from there, scientists have to take additional time-consuming steps to confirm that the molecules are indeed present and bound to the beads. The MIT team found a faster way to confirm the presence of Dynabit-bound pathogens using optics, specifically Raman spectroscopy. This optical technique identifies specific molecules based on their Raman signature or the unique way in which a molecule scatters light. The researchers found that Dynabits have an unusually strong Raman signature that can be easily detected, much like a fluorescent tag. This signature, they found, can act as a reporter. If detected, the signal can serve as a quick confirmation within less than 1 second that a target pathogen is indeed present in a given sample. The team is currently working to develop a portable device for quickly detecting a range of bacterial pathogens, and the results will appear in an emerging investigator's special issue of the Journal of Raman Spectroscopy. This technique would be useful in a situation where a doctor is trying to narrow down the source of an infection in order to better inform antibiotic prescription, as well as for the detection of known pathogens in food and water. Looking for diseased cells and pathogens in fluid samples is an exercise in patience. The numbers present are so small that they must be grown in controlled environments to sufficient numbers, and their cultures strained, then studied under a microscope. The entire process can take several days to a week to yield a confident positive or negative result. Dynabits are commercially available microscopic beads made from a magnetic iron core and a polymer shell that can be coated with antibodies. The surface antibodies act as hooks to bind specific target molecules. When mixed with a fluid such as a vial of blood or water, any molecules present will glue onto the Dynabits. Using a magnet, scientists can gently coax the beads to the bottom of a vial and filter them out of a solution. The research team is investigating ways to then further separate the beads into those that are bound to a target molecule and those that are not. The beads themselves are not visible by eye. The researcher uses Raman spectroscopy as a way to fingerprint pathogens. She has found that different cell types scatter light in unique ways that can be used as a signature to identify them. In the team's new work, they found that Dynabits also have a unique and strong Raman signature that can act as a surprisingly clear beacon. As a practical demonstration, the researchers mixed Dynabits into vials of water contaminated with Salmonella. They then magnetically isolated these beads onto microscope slides and measured the way light scattered through the fluid when exposed to laser light. Within half a second, they quickly detected the Dynabits' Raman signature, a confirmation that bound Dynabits and by inference Salmonella were present in the fluid. The team's new technique is significantly faster than conventional methods and uses elements that could be adapted into smaller, more portable forms, a goal that researchers are currently working toward. The approach is also highly versatile. The team is particularly keen to apply the test to conditions such as sepsis, where time is of the essence, and where pathogens that trigger the condition are not rapidly detected using conventional lab tests.